All right, well, we're going to get started. I'll continue to try to let people in as they come. Welcome, everyone, to New York Sea Grant's Aquaculture Industry Resource Webinars. This one is about the potential role of triploid oyster technology to mitigate disease and increase yield. So just before we begin, I wanted to share some info about the Zoom features and our webinar procedures. I do realize many people have spent enough time on Zoom, but this is meant to help the folks who are less familiar. These icons might look a little different depending on your version of Zoom. You can adjust your mic and video settings by clicking on icons on the bottom left corner. Please stay muted throughout the webinar unless you are speaking. You're welcome to keep videos on or off, but when you're asking questions, it'd be great if you could turn your videos. Questions can be posted directly into the chat box throughout the webinar, so you don't need to remember them until the end. If you want to ask your question, please select the react icon. It'll open up this box usually, and then you select the raise hand. Other versions might just have a hand that you can select. I'll call on you. We will have a Q&A period at the end, but if you have a specific question early on to clarify something that is said by a speaker, you can ask that or put it in the chat and I'll have them address it. But I'd like to hold the general discussion questions until the end so the speakers can get through. So the goal of these webinars is to provide industry information, provide relevant information to the industry that's convenient. Ideally, we'd all get together and have these in-person meetings, but that's not always logistically feasible. So trying to do them online offers flexibility. They get posted on the New York Sea Grant website. You can scan that QR code, and the link is shown there as well. There will be a short survey at the end where you can request topics of interest for future webinars, and also can offer a little bit of feedback on this one. And so today's webinar is to increase awareness about triploid oyster technology, the pros, cons, how the technology can mitigate oyster disease and potentially increase harvest yield. Overall, I hope this will help increase everyone's knowledge about the topics to allow for more informed decisions in the future. So today we are fortunate to have two speakers from Stony Brook University's Marine Animal Disease Lab. Bassem Allam is the director of the MADL at Stony Brook University. He earned his PhD from the European Institute for Marine Studies in Brest, France, and performed his postdoc at Rutgers University in New Jersey before he joined Stony Brook in 2003. His research focuses on shellfish physiology, health, and aquaculture, and has resulted in the publication of over 130 scientific papers and book chapters. And we also have Dr. Brianik, who is a recent PhD graduate from the Marine Animal Disease Lab at Stony Brook. His doctoral research focused on the performance of triploid oysters grown along the East Coast, with particular attention to their size, health, and disease status in comparison to diploid oysters. Chris has published several papers, primarily addressing triploid oyster biology, and his broader research interests include animal health and disease, with a special focus on parasites. So with that, I am going to stop sharing my screen. I'm going to let Bassem take over the screen.
Thank you, Barry. Thank you for the opportunity. First of all, thank you all for being here. While the title talks about triploid technology and oyster, I will leave that part to Chris Brianik at the end, but I will start really by the general consideration of shellfish with a focus on oyster. Most of the example will be on oyster, but shellfish in general, some aspects really apply to all shellfish species, all bivalve species, all marine organisms in general. Yeah, so let's start with that. Basically, these organisms, as you know, they live in that environment. They are exposed to everything that happens in that environment, and we know the environment is changing. Yeah, and each and every one of these elements here can cause at some point, sometimes, not always, luckily, problems. Yeah, and when I say problems, really health problem that can be visible in the tissue, visible in the organism, visible in symptoms, growth, performance, fitness, whatever. Yeah. Most of the research we do in our lab is on infectious agent, but we also touched a bit of everything here, maybe excluding pollutant, I would say. But really, it's not just a disease issue, infectious disease issue, things like toxicology. And you know all that, yeah, you know all that, that these animals are facing all these stressors all at once. And it makes it hard for them, yeah. Luckily, the oyster is rather a robust species, frankly, compared to other species. So it does fine. It grows, as you know, from South America up to Canada. 40 degrees roughly difference in its habitat, 40 degrees centigrade between almost 40 degrees environment and freezing temperature. You know all that. But it faces challenges, and some of these challenges, again, I will not go in detail here. You know all these better than me, frankly. Fouling organism, it's a big issue. What I'm just highlighting is some estimate done by folks is up to 15% of annual operational cost. So this is a problem indeed, and it's always mitigation is better than the solution, because the solution on a problem like this is terrible. It's a lot of cost, a lot of labor. Some of the example of things we see here, and again, you are familiar with all of these. Some of you may have seen these tube-borne that can really extremely difficult to clean. The mudworms are here, the boring sponges. This is actually science in severe cases of boring sponges that you can see even through the shell on the tissue. Yeah, these marks are made by boring sponges. So in some extent, this makes, of course, the shell very brittle, fragile, and hard to work with, hard to sell. And again, you are familiar with these issues. There are some information on these and others. This is one, for example, fact sheet here on Polydora and mitigation of Polydora. You may have access to it. If not, I'll make sure that Barry has the information needed to get it. That gives some guidelines for cleaning these. Even there are one on tube-borne, but it's out of Australia, not over here. Not study not done in the US about mitigation strategies. So our focus is indeed, even the examples I will be giving today are on pathogens. And these, there are many, right? When we say pathogen, most problematic ones are small things we don't see. The big stuff actually that we see are usually, frankly, are less problematic in terms of survivorship of the stock, yeah? But there are signs sometimes that are visible to the naked eye, visible on organismal level. Like if you look at your oyster or shellfish in general, you can see abnormal conditions, and these sometimes are indicative. They are not very specific signs, so let's keep that in mind, that everything I will show here on this page are not specific, meaning they are not linked to a particular condition, a particular disease, but often are linked to problems. So this may be a first yellow flag if it's not a red flag. Yeah. Unusual aspect of the meat, watery, thin, emaciation during period where they should be more fleshy. We know that there is natural cycle of the oyster meat after spawning, for example, where it gets a little more watery. But here we're talking out of range. Yeah. Changing color, color of the digestive tissue, color in the gills sometimes as well can be indicative of problem or not, because we know that the algae also they feed on can change color of these organs. But again, if you have a changing color with a very thin tissue, very watery tissue, that's typically you have already two signs that are in the wrong direction. Yeah. Other symptoms of gaping, again, I'm not talking shelf life here, animals in the field gaping and shrinkage of the mantle, so the tissue retracts a bit sometimes in some diseases. I'll show you pictures. And unusually slow growth, of course, this is often associated with, it's a pre-indicative of big issues, yeah. And of course, the last that nobody wish to get to that point is mortality, because that's mean it's too late, really. The animal lost the battle, and there is not much we can do at that point. Confirmation. So again, as I mentioned, all these are really non-specific signs, and confirmation will require more specific techniques to get to. And so the examples, really, I'm going quickly on some examples that we see in hatchery systems. So vibriosis, infection with vibrio, these are bacteria. Yeah, this is locally seed, large seed, I mean, field farm deployable seed, and above inside are really resilient to vibrios. Yeah, they can accumulate human pathogen vibrios, but not shellfish pathogen vibrios. Yeah, they deal with the shellfish pathogen really well, the bacterial pathogen at that point. The problem happens in hatcheries and in some cases in nurseries. So we are talking really very young animal in which the immune system didn't grow to a degree that they can fight these infections. Yeah? Again, if you are interested in information on human pathogen, we can talk about them later during the Q&A. But here my focus is on those bacteria. Again, I'm not gonna detail in much because most of you don't face these. This is really a hatchery issue, in some cases nursery issue. And it's often, I mean, not always, of course, there may be other problems like algae culture, things like this that are contaminated, but it's often really water management, water quality matters. It's these bacteria in general are what we call opportunistic, meaning in a healthy system, in a good, healthy shellfish, they don't create a problem. They create a problem only when the animal is under stress by some other factor that you need to understand. Yeah. And really improving water quality and good husbandry often resolve the problem on its own. Sometimes you need to empty the hatchery completely, falling, let it dry for a week or two before starting over, but it's not relatively talking. Another bacterial disease that you may see on your farm is what we call juvenile oyster disease, or a more recent name is various oyster disease. It's again, it's a bacterial disease. It's a shell disease. It is discovered. Really, the ground zero for this infection was here in New York, on Long Island, Oyster Bay in particular, in the 90s, late 80s, early 90s. These are signs that are visible to the naked eye. So if you look at the shell here, it's again, we are talking a seed. Yeah? This could be 10, 15 millimeter. I don't have the scale on it, but let's assume it's 10, 15 millimeter. See the cupping here, extreme cupping of the shell. And also the right shell, which is the top shell, it's the right one. Yeah. The left one, the big one on the bottom. The right shell is smaller. You see the edge here. There is a lip from the left shell that is exposed. This is also an indicative of GOD. It's again could be something else, but it's a strong indication of GOD. The real macroscopic, meaning visible sign, is this brown deposit on the inside of the shell. If you open a seed, you will see these signs. This is again, it's a strong indication of juvenile oyster disease. So we are talking here smaller oyster, really below, there is a threshold that is determined by the colleagues who've been working on this, about 20, 25 millimeter, after which the oyster become resilient, very resistant, doesn't show any sign. Range from New York to Maine. It's not south of New York, as far as we know. I never described New Jersey entire. But again, started in New York, so we are the source here. Time of the year, it's summer, really, and temperature. There were lab experiments showing that the temperature matter a lot, and there is no known treatment. So avoid introduction where we can, when we can. Although again, the range now it's New York to Maine, so it's a bit all over the place. And there is some mitigation strategy. There are robust data showing that reducing density of the seed will reduce the impact of the disease. And again, this is a bacteria that is directly transmissible from an oyster to the other. So by reducing density, it's like a human with COVID during the pandemic, those social distancing, it's a kind of similar concept here to reduce the transmission between the individuals. Yeah. And increasing water flow as well. And the good news is that really the disease quickly, animal that survived GOD here in New York became quickly resistant to GOD. So since New York is what we call endemic, meaning the disease, we believe it's always around, the bacteria is always around, without necessarily showing sign, our animals are typically really resistant. Here, animals grown on Long Island is really among the best, I think, in terms of GOD resistance. Yeah. Second one, that is a bigger, I would say, a bigger problem that affects also market size or near market size oyster, so high value product. Yeah. Dermo disease is a Protensis marinus, the scientific name. It's an interesting parasite. It's really first discovery in the 40s in the Gulf of Mexico. Yeah. And then was shown afterwards up to the 90s to lower Chesapeake Bay. Yeah. So it did spread north up to Chesapeake Bay until the 90s. And since the 90s, made a jump through New York up to Maine, pretty much, southern Maine in the 90s. And now, as you may have heard, this year, an outbreak in Canada. Yeah. So it's really migrating with the climate alteration up north. Non-specific signs of the disease, again, things that can be visible on a farm, could be a reduction in tissue mass, emaciation, pale color, changing color in the digestive gland and mantle. Retraction, the mantle retracts a bit. And again, it originates from the Gulf. It's really like warm temperature. And it's funny, there are really robust studies showing that the temperature that limits its spread are not summer high temperature. It's rather cold winter that are becoming milder and milder. And this maintains the parasite to maintain itself. It allows the parasite to maintain itself in the environment, hitting oysters the following year heavily. Yeah. So it's really interesting. It's migrating north with the warming of the winters, apparently. Yeah. Again, the data is solid, frankly, on that purpose. And salinity, it lives really anything between, experimentally, the parasite can live anything between 5 parts per thousand and nearly 45 parts per thousand. Yeah, salinity. So it's a huge range. However, the disease developed better at salinities above 15 parts per thousand. And I know most of our growing farms here in New York are in that area, high risk area, yeah, in terms of salinity at least. It's a summer disease as well. Even though the parasite can survive the winter, the flourishing in tissue and killing the oyster happen in summer, typically September, October. Getting a little earlier now because the spring is getting a little milder as well, warmer. And it worsens with age. So this is what I said earlier, that it's really near market size or market size product. So juvenile, young of the year, they have it, they catch it rather quickly, yeah, but they enter the fall with limited amount of parasite, they survive through the winter, and then the following summer, the parasite take advantage and flourish quickly. Yeah. No known treatment, avoid introduction, but again, we know things move around on their own anyway. Use of resistant oyster. There is, I should say here, the success of breeding for resistance of dermo is way less than success of breeding for resistance against juvenile oyster disease, for example, the bacteria I mentioned earlier, or MSX disease, another parasite I'll be talking next. So here it's more challenging, and the thought is that this resistance may be deriving from multiple genes in the genome. I will not go in details here, but to tell you just that it's really challenging. There is no highly resistant strain. There may be something, some gain in resistance, but it's really not that marked resistance. So it's a problem. There is active research on that in multiple laboratories around the nation. Yeah. So this is another one, MSX. It's a big problem from time to time around here. And this is a picture I took this year. And actually, I took it from a restoration project, not from a farm. But we have it. Yeah, we have it. It shows up every few years, especially when we have less rain. It pop up in the summer, late spring, early summer, when the summer is drier than normal, yeah, or spring for that matter. Could be winter salinity also that are low, precipitation that are low, meaning salinity is high, and the parasite pop up the following summer. So historically in the Lower Bay, and the thought is that it was introduced accidentally from the Pacific Ocean with oyster, with Pacific oyster at the time. Now it's between Florida and Nova Scotia, and they have it in California, West Washington State, in the Pacific oyster, and in Asia, multiple places in Asia. They have it in Pacific oyster as well, which we believe is really the original source of the parasite. Yeah. It's fatal, yeah, but you can have signs of poor tissue, watery adults, changing color. Again, all of these, I mentioned them earlier for another disease, so yet again, it's not very specific, but indicative of a problem. Salinity is really a main driver of the parasite, more so than temperature in this case. I mentioned drought, but again, over 15 parts per thousand, and the mortality typically around the 20 parts per thousand, which again, many of our farms may fall in that area as well. So it can start in spring for infection acquired the previous year. Mortality can happen in spring. Infection acquired in spring, mortality will happen in the summer, that same summer. Yeah. Incomplete life cycle. The previous example I mentioned, we have culture for them. We can culture them and induce disease. Here, there is no culture of the parasite. And there is strong evidence that it requires a yet unknown intermediate host to complete the life cycle, to be able to infect it. So there is no oyster to oyster infection. Yeah. And no known treatment, avoid introduction. But again, resistance here was built up really quickly. I will show you example in a few slides. Yeah, really very impressive how oyster, survivor oyster from infection can resist and pass those genes to their offspring to produce resistant stocks. Yeah. The last example in that theory, I'm going quickly here, it's SSO is a minor, it's similar, it's a cousin of MSX actually, it's real close cousin to MSX. It's like further higher salinities, yeah. The name is seaside organism. The name came from Chesapeake Bay, my recollection. It's shown only on the seaside, on the ocean side of Chesapeake Bay, yeah, of the peninsula, while on the Chesapeake Bay side, we don't have it, yeah. So this is the range, similar to MSX really. In many cases, it's very similar to MSX, high salinity areas. It's less impactful than MSX in our areas here. Yeah. And again, no known treatment. There were work showing that if you move oyster to lower salinities, and here less than 25 parts per thousand, it's not very low, it reduces the impact of the disease and the parasite. Yeah. So again, I will make sure that Barry has access to this website if you have. It's really an amazing website. It's a repertoire of information on these diseases and many other diseases, infecting even oysters, eastern oyster, yeah, that are less important in our area. That's why I didn't list them all. But this website really is a fantastic resource if you want to know more on any of what I mentioned so far. So now I will move to what I believe the more interesting part of the presentation I'm giving here is, instead of listing these diseases, telling you what could matter and how we can mitigate these problems, specifically with selective breeding. Yeah. As a overall concept that you can apply to shellfish, but also to any other organism, yeah. When you have a disease, it's really at the interface or interaction between a few components, one of them being the host itself, yes. And the host, when I say the host, is the strain in particular, genetic background of the animal, yeah. The age, the natural cycle of the animal, and many other things. The environment, and again, dissolved oxygen, pH, salinity, contaminant, the most important, temperature overall, frankly, and the pathogen. So really, you need these three components to create a disease problem, yeah? The environment, we cannot do much about it, frankly. I mean, sometime you can, if you are working in a hatchery, you may be able to play with these, but if you have your farm site in a place, you cannot move it easily, yeah? The pathogen also are out of our control. Once they get here, they get here. Now, if we can reduce introduction, that's good. But if not, well, you have to deal with them. So what we can manage is mostly the host, especially for animal that we farm, we breed. So that's where most intervention can happen. Yeah? I will show you a few examples of research done in our lab that highlight the importance of genetic background. This is a study we did on Cupex disease in clam. This is the example in clam, not in oyster. But we deployed seeds from three different genetic backgrounds, genetic background 1, 2, and 3. We deployed them, we got them the same age, we deployed them in the same place at the same time. And this is disease development for this parasite of clam over time. And the periods are here, the different bars, different period. Strain 1 developed the disease quickly, within weeks, really 6 weeks, and worsened over 1 year. Strain 2 developed a tiny bit of disease. Strain 3 did not develop the disease at all over the 16-month period. Yeah. This tells you that the genetic matters a lot in this case. It does matter also for oyster. And this is an example we have in the lab also. We didn't publish it yet, but it's in GOD, juvenile oyster disease, where we have 4 strains. Each strain show you the cumulative mortality. Yeah. In this case, we exposed the seed to the bacteria that cause GOD, but also in conjunction together with ocean acidification. And you see the mortality. We have 1 strain. They didn't show any mortality. The other strain, we lost over 50% of the seed within a month. Yeah, extremely fatal to this group. And this is a more recent example in scallop studies we've been doing here with Cornell Cooperative Extension, where we produce or we purchase some of these strain, the cohorts, our strain of scallop. Some of them we produced ourselves at the Cornell hatchery. Some of them we procured, and we compare them. And you have up to 4 times mortality in this group than in this group. Yeah. 4-fold mortality within 5-month period. Yeah. So genetic matters, all that to say. And when I say genetic matters, we can have, and we do have the tools to tell apart genetically, based on the DNA makeup, some markers associated with the resistance. Yeah. So here it's 3D schematic representation. I'll not go in detail, but shows you basically animals that survive 18-month period here, yeah, this color, 18 months, are genetically different from animals that succumb or animals that we had at time 0, meaning survivor, yeah, the winners part of the population can be segregated genetically from animals that succumb to the mortality over 18 months, yeah. So these are what we designate our marker of resistance. The same thing with the oyster. In the case of ocean acidification, in this case, again, survivors. In this case, actually, in this case, it's not survivor over time. It's really animal that made it through ocean acidification event. Yeah, the red ones can be genetically distinct from animals that were not submitted to that stress. Meaning, we have a selection, and that selection we can follow it genetically. This is MSX case done by late Professor Susan Ford, who was my postdoc supervisor at Rutgers. It's a 6th generation selected line of oyster. At time 0 in each of these cages, they put the same number of seed. Yeah, at time 0, the same number of seed, selected on the left, control non-selected on the right. And this is the outcome of the field deployment in terms of number and in terms of size. Yeah, the unselected are here, the selected are here. Yeah, 6th generation is here, 7th generation is here. So they grow faster, they grow better, and they survive more. And again, the picture is, I think, very, very telling how selection can happen. We did the selection for clam, and again, here we selected these 3 groups out of the same original population. And one of the selected line we produced survives about 8 times better than this line, for example. Here, frankly, it's a disease level, not survivorship, although there's a negative correlation between disease and survivorship. But 8 times less disease than the control group. And I want to mention quickly, and this is where I will hand it to Chris after the next slide, one more slide. Selection for fast growth, excuse me, has the potential to also reduce disease risk because your crop will be in the field for a much shorter period of time if the animal grows fast. So there is less risk of being exposed, or they may still get exposed, but the parasite, the disease doesn't have time to develop to a fatal level. Yeah. So final slide, really, in what I want to present, general guidelines. Yes, and these are easier said than done. Yeah, let me tell you this to be in all fairness. Prevention prevail, of course. Not many curatives, unless a low salinity environment. Actually, in the past, there were farmers here on Long Island who had the luxury to be able to move their crops from one environment to lower salinity environment for a certain period of time, then bring them back for the final grow out phase before marketing to bring back the salt water and the salinity and the taste. Yeah, many of you cannot. You're stuck with your side. Yeah. Use local selected stock. I know these are limited, but I know also there is effort to improve that. The offer on the seed, local offer on the seed, I think it's really important, and we need to work with that. So use that because these are seeds produced from rootstock that grow in our area. They are adapted to our environment, to our packaging. Yeah. Proactive, yes. Stock and density management. Many diseases are really worsened with high stock and densities. So we have examples in oysters and clam and many. Growing device maintenance. This is also important. Water flow. Again, I know it's labor. I know you try your best to do it. That's really important. Surveillance, when you can really have the luxury of being able to do proactive surveillance on the stars. It's costly, I know, but at least surveillance at the organismal level. Keep an eye on the stock, and you do that. I know that. I'm not giving lessons here. Yeah. And finally, production cycle management. What I mean by that, we know that some diseases, if we have them in the stock, we know for sure that they can worsen in particular period of the year. So that may be a way to manage also the available stock, the growing stock for marketing. If you can sell it before the period that you know for sure, you may start seeing mortality. That's my final word here. Again, I'm happy to take question at the end. Yeah, thank you. Chris, it's all yours. I'll stop sharing my screen.
Thank you, Bassem. And yes, while Chris is pulling up his screen, if anyone's got questions, you can put them in the chat so you don't have to try to remember them. And it's possible Bassem can answer some via chat. But yeah, take it away, Chris.
Thank you, Barry. And thank you, Bassem, for introducing a lot of the concepts I'm going to be talking about in this presentation.
You need to go one more.
Yeah, there we go. Looking good?
Yep.
Cool. Hi, everyone. I'm Dr. Chris Brianik. As mentioned before, I recently finished my PhD at Stony Brook, working in the Marine Animal Disease Lab, where my PhD was focused on growing triploid oysters, specifically around Long Island, but in the Northeast in general, and addressing some of the concerns that farmers have raised about triploid technologies and their limitations or issues they've occurred with them. So that's what I'm going to be kind of talking about in this. I'm going to start real low and start working my way up, bringing in some of my own data as we go. So first, I want to start off with just what are triploid oysters? And to keep it simple, triploid oysters are just oysters that have been specifically bred, have an extra set of chromosomes compared to your standard or traditional oyster that has 2 sets of chromosomes. Now, your standard oyster is bred this way. Generally, you take a male, you take a female, you take the gametes from each one, and you mix them together, and then you get a whole bunch of baby oysters that have the same number of chromosomes. Pretty simple. Triploid oysters are made very similarly, except instead of using a male that has 2 sets of chromosomes, you use a male that has 4 sets of chromosomes, known as a tetraploid oyster. Also, sometimes some people like the term triploid or triploid. I like triploid, so I'm going to be using that one, so don't get too mad if you hear that wrong. So you take a tetraploid oyster and you spawn that with a female diploid oyster. And when you do this, you get 100% triploids. Now, here I listed Rutgers NEH as the tetraploid because in our region in the Northeast, it's really the only line available for tetraploidization. Though down south in like Louisiana and in Virginia, there are a few other tetraploid lines that can be used to make triploids. However, they wouldn't do very well up north because they have different stressors that they've been adapted to. So up north, you're really only going to be seeing NEH as it's really the only established line. And you can make triploids in a few variety of ways. This is the main way that's used for eastern oysters now. You can use chemicals to induce triploids, but really, if you're working with the eastern oyster, this is the only method that you're going to be encountering. All right, so moving on. Now that I explained how they're made, why do people care about them? Is my slide out of order? Slide out of order, that's fine. So as they're made, the reason they're made and why you probably hear about triploids all the time is they have some unique benefits. Being that they have an uneven number of chromosomes, this makes them functionally sterile. They can't produce gametes very well. And because of this functional sterility, they end up having about 30% better growth rates compared to their diploid counterparts. This is typically measured in mass, but in terms of length and width and cupping, these are all advanced as well. On top of this, just more meat, you get more consistent meat quality. So pictured here on the right, you can see a diploid that's going through gametogenesis, all that white developed gonad. However, if you look to the left of it, that's a corresponding triploid of the same age from the same stock, most likely. And you can see that it's relatively just no gonad development really going on there. And as a result, triploids can be sold year round more consistently. You don't have that boom and bust cycle of the spawning with diploids. And I've heard that for some people that have to ship them kind of far, they like working with triploids because there's no risk of them spawning when you're shipping them on ice overnight or anything like that. They stay much more consistent. And on the shelves, they are less likely to spawn, again, resulting in just that higher meat quality and more return customers as a result. Now, due to these main reasons and a few other minor reasons, triploids have become incredibly popular. They account for more than 90% of the oyster spat produced in France. More locally in Virginia, they account for more than 90% of oysters sold, and I believe that was from 2016, that data. Down in the Gulf and on the West Coast, these trends are following in suit. We don't have specific numbers, but I've been led to believe that in the Gulf, it can be anywhere above 70%. And pictured here is just why they're so popular in so many areas. On the bottom, you can see triploids and diploids that respond at the same time, and the triploids are just notably larger than the diploids. Additionally, down south, where they're not concerned about growth rates, the lack of gonad development really just helps them maintain a more consistent quality meat throughout the year, which is really helpful for their product. So, however, Long Island farmers, when I've talked to them, they've given me this warning. They're frail and sickly. They don't do well. They don't perform well. You buy them, they cost a little bit more money to buy, and then they just die on you as soon as you buy the seed. They're just not performing. The reports that other people have displayed are just not aligning with what Long Island farmers have encountered. So this is really the main core of what my PhD dissertation was. It was to address, are triploid oysters on Long Island frail and sickly? Is Long Island just unique in that they don't do well here? Do we have a unique environment that triploids are just doomed to fail on Long Island? Or is this more related to some other options that might be influencing the outside data that could be confounding some of the results? So that's really what we sought out to do during my PhD. But to address this, the first thing we had to do is we had to spawn oysters. Now, to spawn oysters is relatively simple. You take an oyster, a male, a female, you cut the gonads out and you mix the gonads together, and that's pretty much what we did. However, we wanted to produce both triploids and diploids. So what we did is we sourced male oysters from Rutgers. We got both diploids, so your standard diploid, and tetraploids. We would get local female oysters, we'd strip spawn them for their eggs, and then we'd split those eggs evenly in two. We would then fertilize those eggs with either the diploid sperm or the tetraploid sperm, and this would allow us to get both diploid and triploid oysters that were half siblings of one another. And this is important because oysters can have a lot of genetic predispositions that allow them to succeed in certain areas and not in other areas. And different lines are really good at different things. Some are better long-term, some are better short-term, some grow really fast, some have better flavor. So we wanted to control for that as much as we want. So we kept the genetics as close as we possibly could. So this is how we would spawn our oysters. And we did it over 2 different years. We have spawn year 1, spawn year 2, which is 2020 and 2021. And at each one of these spawns, we spawned 6 different oyster lines, 3 triploid, 3 diploid lines. Now, the specific lines aren't terribly important, just showing that we use genetic variety in this experiment more so. And I'm not really going to be focusing on the individual line comparisons because that might not be relevant to everyone here. I'm going to be mostly focusing on just the diploid-triploid comparison, but showing you that we did test multiple genetic stocks and we did it over multiple years so we can really figure out what's going on. Now, as I mentioned, we were told by some local farmers that the issue that they've had with triploids are if they're buying the seed and it just kind of falls off, it's frail, it doesn't do well, it looks weird, and it just kind of fizzles out and they're spending more money for it. So we wanted to start our investigation assessing triploid-diploid dynamics right from the get-go. So that's what we did. So starting at 1 week and 6 weeks post-spawning in the hatchery, we were able to collect larvae oysters and juvenile oysters from each one of those lines, from all 12 lines across both years. We would collect a few hundred to a few thousand of these oysters, depending on the specific experiment. And we bring them back to lab. Then once in lab, we would take these oysters for each line and we'd separate them into 6 replicates. And then half those replicates, we would then expose to pathogenic bacteria. A specific number of bacteria relative to their age class. The older the oyster, the more bacteria we gave them. And the bacteria we used are some of the ones that Bassem mentioned before, mostly Vibrio bacteria. And these bacteria are specifically known to cause hatchery crashes. So they get into a hatchery, they replicate exponentially, and they can just wipe out an entire stock. So these are bacteria of relevance to these age classes of oysters. And what we did is we exposed them, and then we would follow their survivorship over time and see how well the oysters from each one of the lines are surviving compared to their half-sibling counterparts and to the other lines as well. Now, before I start sharing the results of this, I want to bring up another experiment we did. So these experiments only used a few thousand oysters, and we produced tens of thousands of oysters for these experiments. So with the remaining oysters, we did a field study. So we could compare not only these early age classes, but the more relevant to the farmer age classes being the field grow out period. So Spawn 1, or 2020, was mostly focused on Long Island. So the picture to the bottom. Each star represents a location in which we deployed oysters. Oysters were deployed in triplicate. So for each line, we deployed 3 different ADPI bags, stocked at about 200 oysters per bag. And then, sorry, my dog is looking at me through the window. So we stocked the bags at about 200 oysters per bag. And we did this in triplicate at each one of these sites. And some sites we had both floating and bottom gear. So we replicated a few different growing conditions as well. And this was, again, just on Long Island. And top of those 6 oyster lines I mentioned before, the 3 triploid, 3 diploid that were half siblings, we also got an additional 3 oyster lines that are purebred lines. So oysters that were grown at their local hatcheries, that how they typically would, that were donated to us. And this way we could see that our triploids don't only outperform their half siblings, but they are also going to be compared to what's already being grown in the location. That way we know that our triploids aren't only outgrowing their half siblings, but they're also doing better than what's already grown. Spawn year 2 was very similar in general design, though it was spread across the East Coast instead of just on Long Island. So from Maryland up to Rhode Island, again, each one of the lines was deployed in triplicate. And we had a corresponding purebred line at each one of the locations. Now, the purebred line from Maryland wasn't deployed in Rhode Island, for example, because there was 2 different locations. So each location only had 1 purebred local line this time. So slightly different, pretty close. So with that, we can start talking about some of these results. So first, we're going to talk about the survival results. First, I'm going to focus on the bacterial exposure experiments. So on the left axis, or the y-axis here, we have the average survivorship of oysters after they've been exposed to bacterial pathogens. On the x-axis, we have larvae and juveniles. And for both spawns, I combined all the diploid data and all the triploid data into just one data point. The diploids are in red, the triploids are in blue. And you can see for the larval and juvenile oysters, the triploids had significantly lower survival than their diploid counterparts. For the larvae, diploid survived about 50% more than their triploids. And for the juveniles, they only had about 15% advantage, a little bit lower, but still significant. That's what the asterisk sees above the data suggests. Now, so this shows that when they're young, these triploids do seem to be a little bit more frail than their diploid counterparts, and maybe even a lot more frail based on the larvae data, though it seems to be decreasing as they got a little older. So then after the oysters were in the hatchery for about 2 to 3 months, we deployed them in the field, as I mentioned, and then we would check on their survivorship every few months and collect data on their size and all that, and that's what I can present now. So this is the field grow out survivorship data. On the y-axis, again, we have the average survivorship of the oysters. X-axis is how old they are. So at 5 months, they've been in the field for about 3 months. At 12 months, they were in the field for about 9 months. And for 1.5 years, they were in the field for about 1 year, a little bit longer. And their average survivorship on this graph represents how many survived from the previous time point. So at 5 months, about 30% of the oysters that initially were deployed survived to the 5-month mark. At 12 months, about 70% of the oysters that were present at the 5-month mark survived to that time point. So just so you can follow. And again, we put these oysters in specifically stressful locations. We wanted high stress, and that was so we could really tease apart if there's diploid, triploid dynamics. If triploids are more frail, as people claim, the higher stress, the more likely we would be to see those different dynamics. So that's why we're seeing slightly high mortality in these graphs. But what this is really showing is that the triploid and diploid trends remained pretty consistent regardless of location. If I were to break out this graph into every individual location, you'd see pretty much the same trends, just on slightly different extents. But the diploid-triploid dynamic really stayed consistent between all the locations and the time points once they were deployed in the field. Now, I've also collected growth data. So on the y-axis, we have area, that's the height of the oyster times the width of the oyster, and it's in millimeters squared. And on the x-axis, we have the age of the oysters from 3 months, which is when they were initially deployed, to 19 months, which is when they were finally taken out of the water. And we can see that at both spawn year 1 and spawn year 2, or 2020 and 2021 spawns, we see that triploids perform significantly better than both the diploids, and in the green, we have the local purebred diploids here. Sorry, I didn't mention that sooner. There's a lot of variation around some of these data points, but that's just because, again, we're combining a lot of different locations into one data point, so it increases the variation on those bars. But these were significantly improved growth results. Now, you can see the extent of the variation, or the extent of the difference, is a little higher for spawn year 2, and that's just because some of the sites really exploded, and that has to do with the difference in geographical distance, where the spawn year 1 was a little bit more tight-knit, and it was a very strong growing year in general. And you see that the triploids really overtook the local diploids and their purebred half-siblings, or their half-sibling hybrids, around the summer months between months 12 and 16, which is when reproduction was really occurring in the diploids, allowing the triploids to really overtake. All right. Now, beyond just growth, we also measured wet weight. It followed very similar trends, so I'm not going to show you the same data 3 different ways. It's all pretty much the same. But we wanted to also look at disease presence. As Bassem mentioned, disease is a big player in our area, and particularly a little south and a little north for different reasons. So what we did is at the 19th month mark, when we finally ended the experiment and took the oysters when they were adult size, we assessed these oysters for multiple of their parasites. The data I'm showing here is the dermo pressure, or the dermo weighted intensity, or Perkensis moranis. Now, dermo weighted intensity typically goes on a scale from 0 to 5. All the infections that we found were very light, so that's why you're seeing infections all pretty much below 1. And again, I'm combining all the sites within each spawn year, so the data is a little squeezed together in a sense. And what this is showing is that for the diploids and the triploids and the local diploids, they had very similar infection prevalence and intensity. So derma was about the same at all these sites. And in spawn year 2, you see that the local diploids look like they did much better. This is actually just an artifact in that the site that had really high parasite loads in the diploids and triploids just didn't have a local purebred diploid. I can go into the nuance of that. It's just line crossing problems and complications of experiment, trying to summarize it. But it's just an artifact. And what this is actually showing is that the diploids and triploids really have pretty much the same resistance to dermo infections. We also looked for MSX throughout these experiments, and in 2021-ish, there were no oysters with any MSX found in over 600 oysters. Same with the following year. We found some MSX in the New Jersey location, and when we compared diploides, again, there was no difference between diploids and triploids in terms of disease resistance. So we're finding really similar results, and the data from the literature also suggests that they tend to do about the same. But what's this mean when you take it all together? Well, we do find some data suggest that triploids might be a little frailer, but this is really only at the early age classes and shouldn't really be coming up in a farmer's grow out. They should be past that early osteogenic triploid frailty by that point. but I can discuss the specific reasons why you guys might be encountering this in the next slide. Triploids do have advanced growth rates on Long Island, and they're pretty consistent amongst the sites and amongst the different lines. It really doesn't matter what line you used. As long as you control for genetics, you should be seeing advanced growth rates. So if you source your oysters from the same, if you can source your triploids from the same source that you got your diploids, you should expect advanced growth rates regardless. If you source them from different hatcheries, you might be starting to encounter some differences, and that can be something I can talk about in a little bit as well. And lastly, disease was equally present amongst triploids and diploids. This was found in a few other research areas from down south. So if you have high disease burden for dermo or things like that, working with triploids shouldn't be an issue for you in this regard. Additionally, because the triploids do grow faster, you actually might be able to get a little benefit in that you can take them out of the water a month or two earlier and maybe avoid that high mortality season that some of you encounter. Now, on to why there might be some reported frailty amongst triploids. There's three main reasons that I believe that people have been encountering this. One would be seed source. Getting triploids on Long Island can be incredibly difficult. Most hatcheries don't produce them and they don't do it consistently. So this makes a lot of people have to source their seed from out of state if they want to try triploid seed. Seed sourced out of state can be great. It can also not be like cued to Long Island environments. Environments unique on Long Island can be highly stressful for these oysters. Oysters sourced from areas such as Maine might just not be ready for the different stressors we have on Long Island. And if you're getting your diploid seed from Long Island, your triploid seed from out of state, it might just make it look like the triploids are frail, but it's really just not the right seed sourcing. And we have a lot of JOD on our island, as Bassem mentioned before, and this was something we saw consistently throughout our sites, and that the more removed the oysters were from Long Island environments, the more we saw of this JOD, and certain lines were really predisposed to this. However, it was equally shared between the diploids and triploids, so it didn't really come out. Seed age could also be an issue. I know a lot of farmers, triploid seed is expensive. You want to save your money where you can. So if you can buy them a little bit younger, you might be able to save a few pennies. However, by doing this, you actually might be kind of itching into that early osteogenic triploid frailty zone. We saw that the triploids at their earlier age classes are more frail. We don't know where the cutoff is exactly. Is it at 7 weeks or is it at 2 months? Somewhere in between, most likely. So if you're really trying to get the smallest seeds you can, you might be actually inadvertently getting some of the more frail seeds. So waiting a little bit longer might be beneficial for you. And additionally, there could be certain pairings of genetic lineages. I didn't get to talk about this data too much, but there could be certain lines of oysters that are just predisposed to not being great at being triploids. The way we make triploids, again, is we take an outside NEH oyster and we cross it with a local oyster. And some of these local oysters just might not be very good at doing that. Triploidization can be a little stressful on the cells, particularly at the really early age classes. So this might be a problem that this is more for a research focus, not for an application focus. So at this time I can take any questions, and I left my contact information on most of the data that I presented here, if not all of it, was recently published in the paper if you want to read more on that, if you want some more of the in-depth investigation. But questions at this time would be fantastic.
Thank you very much, Chris, and thank you, Bassem. Chris, I'm actually going to share my screen, and that way I'll have both of your contacts up as well. But yeah, so if anyone has any questions, by all means. And then also here's contact information for everyone. I lost my chat window. Yeah, so if anyone has any questions, feel free to pop them in the chat. Chris and Bassem can stay a little bit longer if needed. I know we ran a little long. And there's also on the screen here is a 4-question survey. This will give me the opportunity it'll give you the opportunity to request future topics. Also give us a little bit of review on, did this help provide you with valuable information? And also one of the questions is specifically for New York Sea Grants reporting purposes. So I'm not going to hold you to the fire if you say you do or you aren't going to do something, but it's just for our reporting purposes. There's a lot of information to digest there. Probably could have been 2 separate webinars. Does anyone have any questions? It's been relatively quiet in the chat. Take the awkward silence as no. Maybe you guys covered it all, or maybe you covered so much that a little overwhelming of information there. I will try to put together kind of like a bulleted fact sheet that has information, kind of summarizing a lot of this information. So I'll do that at the end and have this recording posted, probably not till sometime in January. I have to go through all the closed captions, which is slow and time consuming. But yeah. I'll give it another couple of minutes. If folks have questions, feel free to speak up, raise a hand, pop it in the chat. Stop sharing for now. Hopefully everyone was able to grab contact info. If not, you can always email me and I can give you the contact info.
I will add to the chat, Barry, the link for that website that you can add to the bullet point, maybe.
Yes, yeah. I meant to say that a handful of the slides, I don't know if it was just my screen, they were just a hair cut off at the end, at the bottom, so I couldn't see that fact sheet. But yeah,
okay,
you can email me them. Bassem is going to be on sabbatical next semester, so he'll be less available. So that's one of the reasons we wanted to get a hold of him now.
Not really less available. It's funny because the students here were asking, well, we'll have always our weekly lab meetings. We'll always have the same research. Actually, it's not going to change. It's just the teaching, frankly, and the location.
Okay, so you're still around.
The location, I'm not here physically, but the type of work, it's not going to change at all.
Gotcha. Okay. Well, yeah, Bassem just threw that in the chat. I'll make sure that that is included on the website as well, with the posting and everything. But yeah, it doesn't look like there's been any questions, so.
We answered all the questions, obviously.
Hello?
Yes.
Hey, this is Andrew Larkin, Bassem, and Chris. You guys, I have an oyster farm in Southold, and Bassem said a couple times about getting seed locally. Could you just expand on that a little bit? Because that's kind of one of the filters I'm trying to work through is if you get seed from out of state, trying to keep an eye on that versus other seed. And
yeah,
I mean, if he thinks local trumps everything else, that would be helpful.
Yeah. Everything is relative in terms of local, yeah, relatively. If you can get seed from a hatchery in your embayment, do that. I know you will not have many choices. Well, you're not very far from Cornell, maybe if they have access, but they cannot sell. I know that. So locally here means as close as possible, because thinking on a geographical scale, I know many people get seed from really location north, and as some of these stock are really not used to our environment, they may do wonderful in given years, but some summer may be too harsh for them, and disease pressure also is not the same here as, say, in Maine, for example, yeah? So these boot stock are not exposed to the same. Let's talk only disease pressure, yeah? Not exposed to the same disease pressure, hence they may be, if there are uptick in disease levels here in our area, they may suffer more. So local being as close as possible to your environment, location, environment, characteristics, even out of state, that's fine. Yeah, I mean, what is the difference, frankly, between Rhode Island and where you are? Yeah, not a huge. But if you tell me Maine, that's a little further away.
Okay, that's helpful. I guess a question for Chris. Did you try and understand the strength of the shell in the diploid versus or diploid versus triploid?
Yeah, I looked at it a little bit. I weighed the shells. I didn't really test the strength because it can be difficult to uniformly test, like empirically test shell strength from a really rigid scientific standpoint because of the non-consistent shape that you find a lot of the time. And I didn't do any tumbling or anything for my oysters just because my oysters were for science, not for eating purposes. So I didn't have to do that extra work. But I didn't see much difference in terms of shell strength between the triploids and diploids. It seemed to be mostly line dependent when I did see differences. And yeah, I can't answer it much more than that. Things like boring sponge impacted them both equally when I looked at that. And yeah, it's just the way you grow it seems to be more important for the shell strength, in my opinion. Like if you're growing on floating gear, you're going to have better shell strength. Or if you tumble frequently or semi-frequently, that seems to be more important for really building that strong shell than the differences between ploidy. I think they allocate equal amounts of energy to growing their shells. The diploid, triploid. The triploid is just slightly larger when they do it. Did that answer the question?
Yep. No, that's helpful. And then another question: Did you do you guys understand the spawning cycle of the boring sponge? Does that spawn several times a year, or does that recur? Or
Awesome, that's you.
It's beyond my expertise. I would not say something I'm not 100% sure.
Yeah, only thing I can say about boring sponges, when it was grown on the top, it seemed to be much more manageable by the oysters. When it was grown on the bottom, it became much more obvious and impactful. But I think that has to do more with the speed at which the oysters grew. When oysters were grown on floating gear, they grew a good amount faster. And I think they could kind of outgrow the boring sponge a bit. But when they were grown on the bottom, they grew a little slower, and that allowed the boring sponge to get more of a foothold.
Andrew, someone had asked me about that, and I had posed the question in the Aquaculture Information Exchange. I can't recall specifically the answer I got back, but I'll look into that and forward you the information they provided me. I'm inclined.
Yeah, I'm trying to figure out like the optimal time to brine. You know, when you have to brine everything, if you could do it once and kind of knock the boring sponge out for the season, or if it's something you should be repeating every few months. But it's a huge. It takes a lot of time to do, a lot of work and a lot of time to do. So if you can kind of optimize how you do that, that would be helpful.
Yeah, I think that the information that was provided was that they're not sure that it's a one-time spawn, that it might be like kind of a couple of month window, but I'll double check the information and I'll forward that to you, Andrew.
Okay, thank you. Thanks, you guys.
No problem. Any last minute questions? Thank you, Andrew, for breaking the silence. All right. Well, I hope that this was valuable to you. Like I said, if you go take that survey, you can request some additional topics. You can always just email me as well. And with that, thank you, Chris and Bassem, for joining us. And thank you, everyone, for attending.
Thanks, Barry, for arranging this. Thanks
Bye, guys. Bye, everyone.